Welcome to the second video of our series about meiosis. If you haven't watched the first part yet, you should do that now. Simply click on the link here. If you're watching on your phone, you can find the link in the description box below.
In the last video, we explained the basic terms that you need in order to understand meiosis. After going through interphase, the germ cell can now start meiosis 1.
What's meiosis 1?
Meiosis can roughly be divided into meiosis 1 and meiosis 2. During both stages, the cell goes through the phases you know from mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. The outcome, however, is very different from mitosis. During meiosis, the mother cell is not cloned. Instead, the result of the division of this one original germ cell are 4 new gametes. Each of the gametes contains a haploid set of 23 chromosomes, and each of the gametes contains different genetic information.
And how exactly does that work?
We will explain that now. It's going to be complicated. We highly recommend you to watch our video about mitosis first, if you haven't already. In that video, we have explained important terms that we cannot explain again here, for reasons of time. Simply click on the link displayed. You'll also find it in the description box below. Let's begin with meiosis 1. After the DNA got replicated during S phase and checked for mistakes during G2 phase, meiosis starts with prophase 1. In prophase 1, the chromatin fibers in the nucleus condense into 46 double-stranded chromosomes. What follows now is the first fundamental difference between meiosis and mitosis. The homologous chromosomes get together as pairs, the homologous chromosome pairs. After that, crossing over takes place.
Crossing over?
During crossing over, individual homologous gene segments between homologs are interchanged. Similar gene segments of paternal and maternal chromosomes of a homologous chromosome pair are swapped. Crossing over results in a recombination of gene versions, alleles, of the homologs. Paternal and maternal genetic information are newly and uniquely mixed up.
And what is that good for?
Crossing over is one of the processes that guarantee the genetic diversity of our species. Through crossing over, paternal and maternal genes are newly combined. After crossing over, paternal and maternal DNA are no longer separated into different chromosomes. Hybrids, chromosomes that contain both paternal and maternal DNA, have developed. Every chromatid of the double-stranded chromosomes is now different to its sister chromatid. They are not identical anymore. Thanks to crossing over, children are not clones of their parents. Through crossing over, genes and homologs are individually and newly combined. Each chromatid is now one of a kind. The genetic information in the nucleus of this cell is now different to the genetic information in all other cells of the body. Completely new and unique genetic information was formed.
Does crossing over take place between all homologs of the germ cell?
No. The sex chromosomes are an exception. Males own one X and one Y chromosome. The Y chromosome is quite different from the X chromosome. Its size, structure, and genetic information are not similar to those of the X chromosome. X and Y chromosome are not homologous. Thus, there is no recombination between these two. Between the two X chromosomes of a female, however, recombination happens. During crossing over, the homologs of each pair overlap and exchange similar gene segments. After that, the chromosomes stay connected at the exchange points.
Now we can go on to prometaphase 1.
Exactly. Prometaphase 1 of meiosis is identical to prometaphase of mitosis. During prometaphase 1, the nuclear membrane starts to dissolve. The spindle apparatus is constructed. The spindle fibers, microtubules, spread towards the middle of the cell. During metaphase 1, the chromosomes line up in the equatorial plane of the cell. The crucial difference to mitosis is that the homologs arrange as pairs. It is left to chance which of the two homologs of each pair is placed on which side of the cell. In our sketch, the sides can be compared to southern and northern hemisphere of the globe.
So there is a double row of chromosomes in the equatorial plane.
Yes. The centromere of one chromosome is only connected to the spindle fibers of one centrosome. This will enable the separation of the homologs later on, in anaphase 1.
Does that mean that the chromosomes stay intact and the sister chromatids are not separated?
Exactly. In anaphase 1 of meiosis, the homologs are pulled in opposite directions. Which chromosome moves in which direction is random. It depends on the cell site that the chromosome has been placed on before.
The difference between anaphase 1 of meiosis and anaphase of mitosis is that during anaphase 1 of meiosis, not the sister chromatids but the homologs separate.
Correct. Admittedly, this is complicated. You might not get this right away, so just rewind the video a few times until you understand. Let's continue. At the end of anaphase 1, the cell is elongated with the help of the spindle fibers. Telophase 1 follows. The spindle apparatus begins to decompose. The nucleus at each centrosome is developed. The chromosomes uncoil to chromatin fibers. In the following cytokinesis, the cell is divided at the equatorial plane. 2 cells form. These 2 cells have different genetic information due to crossing over in prophase 1 and the random arrangement of the chromosomes in metaphase 1. During meiosis 1, the diploid set of chromosomes, the 46 chromosomes of the mother cell, were distributed between 2 cells. These now contain a single, haploid set of 23 double-stranded chromosomes. Those chromosomes are new combinations of the original chromosomes.
And what happens now?
We have now finished meiosis 1. Meiosis 2 follows. Meiosis 2 starts with prophase 2.
Wait, doesn't the DNA have to be replicated beforehand?
No, only the centrosome has to be duplicated so that the next division can take place. This is the second big difference between meiosis and mitosis. The chromosomes are not duplicated again between meiosis 1 and 2. At the end of meiosis 1, there are 2 haploid cells which each have 23 double-stranded chromosomes. During meiosis 2, each of these cells divides into 2 haploid gametes with 23 single-stranded chromosomes per cell.
And why is this second division even necessary?
Let's imagine meiosis ends after meiosis 1. In that case, the gametes would contain 23 double-stranded chromosomes instead of 23 single-stranded chromosomes. If 2 of these gametes fused, the resulting cell would have 46 double-stranded chromosomes. But a somatic cell of a healthy human being can only contain 46 single-stranded chromosomes before DNA replication.
And why does it matter if there are 46 single-stranded or 46 double-stranded chromosomes before DNA replication?
If all somatic cells contained 46 double-stranded chromosomes, these would double to 92 double-stranded chromosomes during DNA replication in S phase. In reality, a normal somatic cell contains only 46 double-stranded chromosomes after S phase. What do you think would happen if a germ cell with 92 double-stranded chromosomes went through meiosis?
During meiosis 1, the homologs of each pair are separated. After meiosis 1, 2 cells with 46 double-stranded chromosomes per cell would exist.
Exactly. And if meiosis ended here, these 2 cells would be gametes. If such a sperm would fertilize a similar egg cell, the outcome would be a somatic cell with 92 double-stranded chromosomes. During S phase of interphase, the DNA of this cell would replicate and the cell would now contain 184 double-stranded chromosomes. I think it's clear that it cannot work like that. The number of chromosomes in our somatic cells has to be constant. This is accomplished by meiosis 2. After meiosis 2, the newly formed gametes have 23 single-stranded chromosomes. If such a sperm and such an egg cell fused, the resulting cell would have 46 single-stranded chromosomes. This cell will split mitotically and make up an entire organism.
So the function of meiosis II is to separate the chromatids of the 23 double-stranded chromosomes.
That's right. This is exactly what happens during meiosis II. The procedures of meiosis II are essentially the same as the ones of mitosis. In prophase II, the chromatin fibers condense into 23 double-stranded chromosomes per cell. The chromatids of a chromosome are not identical due to crossing over in prophase I. No two chromatids are exactly the same. In prometaphase II, the nuclear membrane starts to dissolve. The spindle apparatus develops and the spindle fibers start spreading towards the center of the cell. In metaphase II, the double-stranded chromosomes are arranged in the equatorial plane. In anaphase II, the spindle fibers separate the chromatids of the double-stranded chromosomes. The single-stranded chromosomes are pulled in opposite directions towards the respective centrosome. The cell is elongated. In telophase II, the spindle apparatus begins to dissolve. Nuclear membrane is formed around the chromosomes on each side of the cell. The 23 single-stranded chromosomes of each cell site uncoil to chromatin fibers. During the following cytokinesis, the cell is divided at the equatorial plane. 2 haploid cells are formed. These cells each contain 23 single-stranded chromosomes. What is more, the cells contain totally different genetic information due to crossing over in prophase I. Obviously, both cells that were formed during meiosis I undergo the procedure of meiosis II. Thus, there are 4 haploid gametes at the end of meiosis II. Each gamete contains 23 single-stranded chromosomes. All 4 cells contain different genetic information.
We tried to explain the basics of meiosis to you. Now we want to summarize what we've learned. We also want to illustrate the difference between gamete formation in males and gamete formation in females. The germ cell is diploid. In males, this germ cell is called primary spermatocyte after the DNA was replicated during S phase. In females, it is called primary oocyte after DNA replication took place. Here we see the germ cells after their DNA replication. The genetic information is recombined through crossing over. After that, the homologous chromosome pairs line up in the equatorial plane. The homologs are now separated from each other and are pulled to opposite cell sites to the centrosomes. What follows is cytokinesis. 2 haploid cells have been developed. In males, these cells are called secondary spermatocytes, in females secondary oocytes. As you can see, these haploid cells are the same size in males. In females, however, the cell content is not equally distributed. One cell is bigger than the other, but the nuclei of these 2 cells contain the same number of chromatin fibers. Now meiosis II begins. The function of this process is to separate the chromatids of the double-stranded chromosomes. The procedures of meiosis II are identical to those of mitosis. The only difference is that the DNA is not replicated before meiosis II. During anaphase II, the chromatids of the double-stranded chromosomes are separated and pulled to opposite sides of the cell. After cytokinesis, a total of 4 gametes have been formed from the 2 cells formed during meiosis I. These gametes all have different genetic information. They each contain a haploid set of 23 single-stranded chromosomes. The sketch shows that the cell content of the secondary oocyte has again not been equally distributed. 3 small haploid polar bodies were formed. In humans, these don't have a purpose and dissolve. Only the 4th and largest cell remains. This is the ovum. In males, all 4 spermatids have the same size. They will mature to spermatozoa. During fertilization, sperm and egg cell fuse. The fertilized egg cell is called zygote. The haploid set of chromosomes of the sperm is added to the haploid set of chromosomes of the egg cell. The resulting zygote has a double, diploid, set of chromosomes. It contains 2 × 23 single-stranded chromosomes, so 46 chromosomes. From this single cell, an entire organism is developed through mitosis. Yes, you've heard me right. Every single one of you has developed from a single cell. On this note, we want to end our video series about meiosis.
We hope you liked it and learned something new today.
If you have any questions or concerns, please let us know in the comments below.
If you enjoyed watching, please click like and subscribe.
See you next time.
Bye!